Okay, B cell fundamentals again. So with B cell fundamentals, you have your DNA differentiating, you have your nurse cells presenting self-antigens because you want to make sure that your body does not react with these B cells that are being created. So if it passes the negative selection test by the nurse cells that are presenting those antigens, then they will start to circulate throughout the body. So they're immature at this point, but they mature upon finding that foreign antigen to activate themselves. Okay. T cells are a little bit different from B cells in that T cells, one, they're not free-floating T cell receptors. It's not like how with the B cell, it's secreted from a plasma cell that kind of loses the receptor goes off into the system to bind to something, versus a T cell is it's just there, it's got a receptor that doesn't float away. And then the B cells are just randomly looking for something, and if they encounter it, and if they encounter their antigen, they can become activated and do whatever needs to be done. Versus a T cell has to have a major histocompatibility complex and an antigen presenting cell present the antigen to it. So those professional antigen presenting cells could be dendritic cells, they could be macrophages, mast cells, things like that. CD8 T cells are your cytotoxic class 1 T cells that bind with MHC class 1 because it's to detect a virus-infected cell. So if the virus has some form of antigen that infects your, if the virus infects your cell, your antigens kind of are a little bit different. So then your T cell will pick up on that and just be able to induce apoptosis. Versus CD4 MHC class 2 cells are your T helper 1 cells or your T helper 2 cells. So with T helper cells, the idea is they can bind to, let's say, a macrophage, they can bind to a B cell, like an antibody, and then from there they can induce some form of cell death. So they could kill bacteria or other things like that. What's important about CD4 type 2 is that they bind to exogenous peptides, so things that came from the environment. So bacteria from outside of the cell came in, not natural to your system, versus type 1 is endogenous peptides, so it's your cells themselves that is getting detected. T cell development in the thymus. So remember, the thymus is where you develop your T cells to begin with, and you want to make sure that you can save your placenta, fetus, testes, ovaries, eyes, brain, and thymus from circulating immune system, because those sites are important for sexual development, for just overall development and everything like that. With positive selection of T cells, one, you want to check, does it bind with an MHC, a major histocompatibility complex, or you can have negative selection. So do you bind too tightly, or is it self-reactive, basically? And so it has to have both this positive and negative selection to move on to the next phase. You can also have what's called a superantigen, and that might be associated with Staph aureus. So Staph aureus is able of inducing toxic shock syndrome. So the antigen will kind of more permanently link the T cell receptor with the MHC class 2 molecule so that it's constantly inflamed. Okay. Then we move on to the hypersensitivities. So with the hypersensitivities, you've got 4 types. Type 1 is more for allergies. Type 2, you've got these, immune reaction with IgG, and this can be autoimmune kind of things like that. Type 3 is these immune complexes that form because, let's say, for example, your immune system is attacking a cell mass, and then everything kind of clumps together, and then those complexes are actually attacked and targeted. And then type 4 is kind of the miscellaneous, things associated with T cell receptors. So So how do T cells like overreact to something? And so within type 1, again, it's allergies, anaphylactic shock, rhinitis is like the cold. Type 2 is different autoimmune kind of conditions, and sometimes with type 3. And then type 4, again, you can have serum sickness, you can have celiac disease, you can have other things like that. Or you can have Hashimoto's thyroiditis. But there's like another chart in this unit that you can add in.
To go back to the different types of T cells,
Yes.
the CD4, like
Yes.
T helper 1, T helper 2.
Yeah.
What was the difference between T helper 1 and T helper 2, like what they
I don't, you won't need to know that for this exam, but it just has to do with like what specifically are they binding to, or like how they specifically are killing in the cells, and you'll get more into that with immunology.
Okay.
So, in 2 years. Cancer. So again, this chart I think is pretty helpful to tell you the percentages of like how many people die or get this type of condition. So in males and females, if you break it down by sex, then they get their sex-specific cancer at the highest incidences. But lung cancers typically kill the most, followed by those sex cancers. Pancreatic cancer, though, has the highest kind of like overall ratio of like incidence versus death, because pancreatic cancer is typically found very late stage. And then the breakdown continues. So with cancer, there's 6 hallmarks that are very important. Remember that a cancer has to accumulate all 6 of these before it's completely self-sufficient, completely cancerous, completely able to form as its own entity. But typically, as soon as you accumulate like 1 or 2 of these, the process starts to accelerate, because like you kind of increase the probability of more genetic mutations. And remember, cancer is a genetic disorder. It's a genetic disease that you can have familial predisposition, or it can be completely random. And it's like by virtue of age. It's like an age rate related genetic disease. So to go through all of them, so self-sufficiency and growth signals. So that has to do with a mutation in an proto-oncogene called RAS, R-A-S. And so RAS is responsible for switching off the system, this G protein coupled receptor. So when you have RAS, it will be able to turn off the system by hydrolyzing the GTP molecule. And when you break apart the GTP molecule in the system, you can like take it off from its substrate, so then you can stop further signal transduction of those growth signals. So if you have a mutation in RAS, and so it becomes oncogenic, then the cell will always remain active. Okay, then the cell will remain always active, so it's constantly growing, so it's self-sufficient in growth signals. Insensitivity to anti-growth signals, on the other hand, though, you can have, this has to do with cell cycle regulation, and you have a protein called retinoblastoma. And so RB prevents cells from going into the next phase if they're not ready to. And so if you have a mutation in this tumor suppressor, then you need 2 copies to make sure that you've knocked out this like guardian of cell cycle regulation checkpoints. You can have, if you have familial predisposition, you are 100% more likely to get the cancerous form, because it'll be like very likely that you get that next mutation, versus sporadic, it's pretty unlikely that you will get 2 hits in the same gene. Evading apoptosis, so P53 is the guardian of the genome. It is largely responsible for picking up and signaling to the cell to stop cell cycle regulation at DNA damage, and it will kill the cell. If it will cause apoptosis if the cell can no longer repair that DNA damage. And so if you have two bad copies of P53, then DNA damage goes unchecked, and you start to pick up a lot of mutations. So almost like a very high percentage of cancers have mutations in P53, which like drives their progression. You can have limitless replicative potential once you get telomerase. So remember, telomerase has to do, telomeres are like the little ends of your DNA, and they shorten each time you replicate because you can't like you can't completely replicate your DNA without cutting off like a little snippet or like having them kind of messed up a little bit. And so telomerase will build back the ends so that the cell itself can continue replicating. You can have vascular endothelial growth factor, which will stimulate angiogenesis, which is when blood vessels will start to feed into the cancer. Because remember, cancers require high amounts of energy, and so they're constantly like feeding off the host, and so that can lead to conditions like cachexia, which is like that picture of the individual who's kind of wasting away. So it's like they're losing a lot of nutrients to the cancer. And lastly, metastasis has to do with how well a cancer can first detach away from its primary tumor site. So it will have to like understand that like a part of it is going to branch off, and once that part branches off, so it invades into the vasculature, it will start to circulate. It'll transport to distant organs like the lung or liver or bone and like other parts. And once it gets to the new environment, it has to be able to extravasate, so it has to get into a new microenvironment. So like a breast cancer, for example, if it metastasizes to the lungs, let's say, then that cell that was conditioned to live in the breast has to now learn that it has to adapt to the lung environment. And if it can successfully establish itself in this new microenvironment, then you can start to proliferate and you have successful metastasis. Okay. And then, so bone can be associated with breast and prostate cancers. Liver and lungs are typically the most common sites for where these cancers go to. And then type 4 collagenase is associated with like that like spread with the being able to invade other cells and things like that, because it'll break down the collagen in like the different environments. So E6 and E7 are proteins associated with HPV. So remember, HPV is one of only a small handful of viruses that are well characterized into causing cancer, like a causative agent. So it does that by, one, neutralizing retinoblastoma. So remember, you now have unmanned checkpoints. And two, E6 will bind to P53 so you can evade apoptosis. So HPV 16 and 18 are the ones most associated with cervical cancer, and then 6 and 11 are associated with genital warts. You can have foot warts, things like that. So warts, again, are not neoplastic, they're not cancerous, whereas 16 and 18 are cancerous. So now moving on to the endocrine system. So remember, with the endocrine system, it's all about your brain, your hypothalamic pituitary axis, sending signals to secondary to the organ to release some form of hormone. So you've got the anterior pituitary, which is responsible for flat pig. So flat pig again, FSH, LH, ACTH, TH, prolactin, and GnRH. And then you've got the intermediate pituitary, and then posterior pituitary, which is for ADH and oxytocin. Your thyroid has thyroid hormone, calcitonin, parathyroid hormone. Adrenal cortex is pretty important because you've got aldosterone, cortisol, or weak androgens. And then the adrenal medulla is for epinephrine. Pancreas is for insulin. Ovaries are for, and testes are for those more defined sexual hormones. So with the HPA, you have the primary signal from the hypothalamus, telling a specific target organ, you need to secrete your hormone. So TSH, thyroid stimulating hormone, stimulates the thyroid to make T3 and T4, thyroid hormone, T3 or T4. ACTH will tell the adrenal cortex, adrenal corticotropic hormone, to release cortisol, for example. GnRH stimulates FSH, LH production, which will tell the ovaries and testes to produce estrogen and testosterone. And then growth hormone stimulates the liver and the tissues, so it stimulates the tissues to grow directly, but it also it also tells the liver to produce insulin like growth factor 1. And lastly, prolactin will stimulate the breast. So with your thyroid hormone, you've got T3 to T4 levels, and thyroid hormone is good for growth, maturation, cell metabolism, heat production. Basically, it's like everything for like general metabolism, how you can think of it. And remember, what's important are feedback mechanisms. So if the target organ is not producing enough, the hypothalamus will signal the target organ produce more. If it produces too much, it feeds back on the hypothalamus to tell it to stop telling it to do more. Parathyroid hormone is to increase the total amount of calcium in the cell. So you will stimulate the intestines to increase more calcium. You can tell the kidneys to reabsorb more calcium. You can tell the bones to, you can break down bone to release more calcium into the system. You also stimulate vitamin D synthesis to the active form. So all of that makes more calcium. Calcitonin, on the other hand, is the complete opposite. Typically, though, you'll regulate things by PTH secretion, so you'll make PTH to bring up the calcium levels. If you have too much calcium in the system, then you can decrease the parathyroid hormone, or you can secrete calcitonin to get all this set. With your adrenal cortex and everything like that, what you want to do is you have three layers, each responsible for secreting something different. So the glomerulosa, so that outer portion, kind of here, you can secrete mineral corticoids, so aldosterone, which is for that sodium retention. Zona fasciculata is the next phase under. That's for cortisol. Cortisol for stress and carbohydrate metabolism. Zona reticularis is for those weak androgens like DHEA and that's sex characteristics. And then the medulla is the inner portion of the adrenal cortex, which secretes the catecholamines, epinephrine, norepinephrine, which are not steroid hormones. So this is pretty much what I just talked about, but aldosterone is important for the renin-angiotensin-aldosterone system. Glucocorticoids stimulated by ACTH, adrenocorticotropic hormone, are more for awareness, sleep habits, anti-inflammation, carbohydrate metabolism, things associated with stress, basically. And so now we move on to pituitary pathology. And so all of this is more towards first identify like what is the condition. Do you have too much, so hyper, or too little, hypopituitarism? And then is it the anterior pituitary versus posterior? So posterior pituitary is a lot easier because there's like only two different things that are secreted. So one is diabetes insipidus. If you have too little ADH, so ADH is antidiuretic hormone, which prevents diuresis, prevents like secretion of urine. So you could have a problem with either neurogenic or nephrogenic. So neurogenic is like you don't produce it in the brain. Nephrogenic is you don't respond to it with the kidneys. You can have inappropriate ADH secretion with SIADH, and that would be the exact opposite. So hyper secretion of ADH. and then that concentrates your urine. Hypopituitarism, you can have pituitary infarction or panhypopituitarism. So typically you'll have some sort of adenoma, so some sort of cancer in the anterior pituitary. Some of the times it can be null cells, so it has no hormone that it secretes, that instead the space that it takes up takes away from normal cell function in the brain. If you have some form of, sometimes it's the cancer is like right in the middle of your like eye system, or if you have stroke, you can have problems with like cutting off how your eyes are seeing. So remember when we talked about how your left eye corresponds with like your right brain, like left side of the body corresponds with right brain, right side of the body corresponds with left brain. So your eyes actually have like crossing over at the optic chiasm. So depending on where you cut off vision, like where is that blockage or occlusion or something like that, that's how it affects how your eyes will actually see. Hypersecretion of growth hormone can lead to acromegaly if it's during childhood, or gigantism for children. And then hyposecretion can be pituitary dwarfism. So remember, achondroplasia is another form of dwarfism, but it's genetic. Prolactin, that can lead to problems with amenorrhea, galactorrhea, hirsutism, osteopenia. In males, it can lead to hypogonadism, impaired libido, and like other things like that. Same with the thyroid pathologies. It's like what happens if there's too much versus too little. So I would recommend just running through these charts by yourself. So look at if there's too much like stimulation from the brain versus how much is secreted by the actual hormone, by the actual organ itself. And then from there you can start to derive, is it too much being produced because of a cancer? Is it too little produced because of, let's say, Hashimoto's thyroiditis, where you attacked the thyroid so you can't like sense the TSH? You could be problem with Graves' disease. So sorry, I mixed that up. So thyroid dysfunction with thyroiditis, Hashimoto's thyroiditis, is antibodies against thyroid proteins, versus thyroxine production is no longer, with Graves' disease, you have antibodies against TSH receptors. So you have elevated T3, T4, decreased TSH. And then lastly, pituitary dysfunction, so decreased in both counts. I would run through these different symptoms like on your own, but like hyperthyroidism is like too stimulated kind of, versus hypothyroidism is more like toned down, I guess is how you can think of it. And then you can have iodide deficiencies that can lead to goiters. And then again, run through how parathyroid plays a role in all this. So where is the cancer forming? Where is it the kidneys that have a problem? Things like that. And then adrenal cortex problems, hypo again, or like which one are you hyper of? Like which hormone are you secreting too much or too little of? Cushing's disease is too much cortisol, so it can be due to a tumor in the pituitary that tells the cortex to secrete a lot. It could be a tumor inside the cortex itself that produces the cortisol a lot. It could be ectopic, or it could be drug-induced iatrogenic. And then Conn syndrome has to do with hyperaldosteronism. And then you can have adrenogenital syndrome, where you have deficiencies with T21 hydroxylase, which is important for cortisol, aldosterone system, and other of the secondary sex characteristics. And then lastly, Addison's is deficiency in all three. And lastly, if you have pathology with the adrenal medulla, so you produce too much epinephrine or norepinephrine, then you can have pheochromocytoma, which makes you extremely shaky, extremely overstimulated. So that's it for section 1, and so we'll take a quick 2-minute break.